All right, good afternoon, everybody. Thanks so much for joining us. I'm Shana Kelley. I'm a faculty member in the Leslie Dan Faculty of Pharmacy at the University of Toronto, and I'm happy to have the opportunity to talk to you today about the diagnostic tools that we're using to combat the COVID-19 pandemic, and as well to talk you through some of the new solutions that may be needed to combat this global healthcare crisis. So I'll share my screen and take you through a presentation. And I trust somebody will let me know if there's any AV issues, but it looks good to me, so I'll just get going. So in this presentation, it'll be about 30 minutes. And I'll take you through just an overview of where we are with COVID-19. I'm sure that many of you tuning in are very much up to speed on what's happening. I'll then take you through how diagnostics are being used for pandemic management at present. And as I talk things through with you, I think it'll become very apparent that there are quite a few challenges that are presented by the current diagnostic tools that we have to work with. And so this will then bring me to the need for new technologies that are more effective when facing the type of global health crisis that we're dealing with right now. And then I'll also, in the last part of the presentation, highlight some really exciting progress that's being made at the University of Toronto in this area. And then we've left plenty of time for your questions. So please, as the presentation proceeds, jot down your questions, put them into the Q&A interface, and we'll have lots of time to go through them. Okay, so I'll start with just some general things about the COVID-19 pandemic. As I'm sure you're all aware, this is a pandemic that has been caused by the SARS-CoV-2 coronavirus. The scale of the COVID-19 pandemic is really quite unprecedented. It has affected a very significant number of people worldwide with a really staggering death toll. And in addition to this being a global health crisis, it has also precipitated an economic crisis. So as we've had to shut down as a society to prevent further spread of the virus, we've seen the economic implications that come along with those shutdowns. And we've seen some of the highest levels of unemployment ever recorded. And then, of course, all of the associated financial impact that comes along with the loss of jobs and an unsteady economy. Also very distressing is the impact of the COVID-19 pandemic on vulnerable populations. So we've seen the pandemic hit minority groups very hard, and in some parts of the world, rates of infection are higher in minority populations than the general population. And certainly across the globe, the economic impact of the pandemic has been really severe in these vulnerable populations and for minorities. In Canada, where we've struggled the most perhaps, and our most vulnerable population seems to be the aging population and those who are in long-term care facilities. So over 80% of the deaths that we've seen happen in Canada have been individuals housed in these long-term care facilities. And so clearly, while there's been a really heroic effort around mobilizing as many resources as we can to contain the pandemic, there's more work to be done. And so I'll start off by just talking a bit about how diagnostics are used, especially in the context of a viral outbreak like COVID-19. And they can be used in a variety of ways. So if we're able to detect the SARS-CoV-2 virus in an individual or in a population, that can be very informative, not just for personal management of infectious disease, but also to really understand how the population is doing with rates of infection as a whole. And so Screening large groups of people is very important in terms of understanding the spread of something like the novel coronavirus. We also need to be able to diagnose individuals very specifically and accurately in order to identify people that have been infected. And then for individuals that have come into a clinical setting, let's say a hospital, it's very important to be able to monitor these patients serially, to understand how the infection is progressing, and to ensure that we're getting patients the best possible care that will give them the best outcome. We have a variety of diagnostic tools on hand at the moment. There are two main types that are currently in clinical use. One type detects the virus directly. And these types of diagnostics typically look for the RNA genome of the SARS-CoV-2 virus. And these types of diagnostics then really report on active infections. Does the sample that's been taken from a patient contain potentially infectious virus? There's another type of diagnostic that looks for antibodies, typically in the blood of a patient. And these are indicative typically of a prior infection. So your immune system, as a viral pathogen enters the body, the immune system starts to produce these antibodies as part of their response, and then they linger for quite a long time to provide immunity, and they're also then markers that an infection has happened. Most of this presentation is going to focus on diagnostics that are used to look for viral RNA and diagnostics that are used to identify active infections. And so I'll tell you about the mainstream type of testing that has been really the workhorse thus far in the COVID-19 pandemic. And this type of testing relies on an approach that we refer to as PCR. That's the polymerase chain reaction. And so how this works is that a nasal pharyngeal swab is collected. That swab, if someone is infected with SARS-CoV-2, then the viral nucleic acids would be extracted, and then they're analyzed using PCR. So the way that this then is turned into a test result is that because of the amplification that's provided by PCR, the levels of viral RNA are elevated to a level that they can be read out using light, basically, as a detector of the presence of the viral RNA. So this method, it's very sensitive, it's very specific, it is a really good workhorse method. PCR, though, was invented in 1983. So this is a 37-year-old method. 1983 was around the time that the first personal computers were being adopted. That's when PCR came on the scene. And as you'll see from the rest of the information in this presentation, we haven't necessarily gone past this. So we're still parked in the era of the first personal computers. There has not been an iPad or a smartphone. We're still here in the early 1980s. And so as we've used this type of method to diagnose people with coronavirus infections, a number of challenges have been presented. One relates to the sampling that we've been using. So we use nasopharyngeal swabs so that we have the best chance of collecting as much material as possible from the nasopharyngeal tract. And in order to take one of these swabs properly, you actually need quite a bit of training to do this. There have been studies that have been done on self-sampling, and it turns out that most people are not able to get a good sample from themselves. And this relates to the fact that the swab has to go way back in the nasopharyngeal tract. And again, it takes somebody with quite a bit of training to get a good sample. Once these swabs are collected, they are sent to testing labs. And what happens here is that there's a multi-step protocol that's carried out. In order to run these PCR-based tests, you need a significant amount of training. So all of the technicians that work in these facilities are highly skilled, have had quite a bit of training. And that's really to minimize the number of false positives and the number of false negatives. As well, the facilities in which this type of testing is done have been through an accreditation process to really ensure that the testing results that come out of these types of labs are trustworthy. And so once the tests have been run, a test result is returned, and the result goes back to the patient or a physician 1 to 2 days later. And that's really best case scenario. When the volume of testing really picks up and the labs are very busy, it can start to look more like 3 to 5 days. So this is one of the first challenges that this type of testing presents. It's slow turnaround time. Because we have to transport samples, they get batched, they get run, and it takes a few days. In the meantime, a patient that has been tested who may be asymptomatic or thinks they're asymptomatic may be spreading the virus. So I think turnaround time is a significant issue with this type of testing. And as in Ontario, as the pandemic ramped up, we then went to ramp up our testing labs and found that there were a variety of issues. And the ramp up was actually quite slow, as it took a while to really establish the amount of capacity that was required to process the samples that were coming in. And even more problematic was the supply chain. So in order to run a PCR-based test, there are a variety of reagents that are required in order to work up the sample and then to also be able to run the test. And those reagents were in very short supply because of issues with the border, because of the acute demand for these reagents, many of which are made outside of Canada in geographies or in countries where there was a huge demand locally. And so there were not large amounts of reagents to ship to other countries. So that is one issue that takes time to establish this type of capacity for PCR-based testing. And then once the capacity is established, it is limited. So in Ontario, we ramped our testing capacity. Again, a heroic effort. We went from 0 to many thousands of tests in a very short amount of time. But what we found is that we're really maxed out around 20,000 tests per day. It's going to be difficult for us to get past that. There's only so many accredited labs. There's only so many lab technicians with the right set of skills to be able to run this type of test. And in the US right now, in geographies where viral infections are really spiking, this is becoming a problem. There's not enough testing capacity to keep up with rates of infection. So this is one key set of limitations with this type of lab-based testing, in that there are finite testing volumes that can be realized using this approach. Another significant issue is the way that we collect samples from individuals that want to be tested. So as we're shutting things down and asking people to stay at home to limit transmission of the virus, we ask people who think they need a COVID-19 test to leave their homes and go get tested. And this happens in some places in a way that does keep people isolated. People line up in their cars, but there are significant logistical challenges with this because the cars pile up. There's lines that go on for miles and miles, and people wait in these lines for many, many hours or an entire day. And these may be people that are sick. Maybe even more problematic in Canada, where fewer people have cars, and in the big cities, we don't really have places for these pop-up clinics. People lined up on the sidewalk. So this was March in Montreal when the temperature was hovering around 0 degrees, and we have people lining up, queuing up on the sidewalks, waiting to be tested. And so again, the requirement for people to come out of their homes to get tested for something that is highly infectious that we really want to keep contained, I think is another issue with this type of testing. There are some newer testing systems that solve the problem of the laboratory and technician piece of things. So there are so-called point-of-care testing systems that automate the testing process. So these are small pieces of instrumentation that can be used in a doctor's office, in a pharmacy, anywhere that a sample needs to be processed. And they automate the entire process so that you don't need a testing technician. And there's a company in the United States called Abbott that has produced a new test for coronavirus in an automated system that can be used at the point of care. There's a Canadian company called Spartan that has been working on launching an automated point of care system for COVID-19 testing. A company spun out of my own lab, Exogenic, that was acquired by the US defense contractor General Atomics, also developed one of these systems. And these systems are very powerful because we can then do remote testing. We can take testing out of the lab, but the throughput is so low that it really can't make an impact given the testing volumes that we're dealing with right now. Every sample is processed one at a time, serially. Every sample needs 20 to 30 minutes before the test is finished. And so that low level of throughput does not necessarily make this type of testing a solution for the problems that we are dealing with presently. And so, you may be asking, why is this all that we have? Why haven't we gotten past the personal computer of diagnostic testing? And why don't we have something that looks like other types of technology? I mean, we have self-driving cars. We have almost infinite computing capacity. We can visualize single molecules. Why can't we do a better job on this and bring more sophisticated technology to bear on this very important problem? And there's a variety of reasons for that. I can't answer them all in this 30-minute presentation, but I'll talk to you about one of the issues. Much of the innovation and technological disruption that happens when we're looking at problems at the scale of the COVID-19 pandemic happen in startup companies that are backed by venture capitalists, right? Those are where many of the new innovations come from. It turns out that in diagnostics, there has been a pretty limited level of investment. If you look at the investment in diagnostic startups versus therapeutic startups, there's a pretty big divide there in terms of how much money goes into these companies. And this really relates to the appetite of investors for coming into these types of companies. And so these are some excerpts from an article on Forbes.com penned by Bruce Booth, who's a venture capitalist. He's a partner at Atlas Ventures in Boston. And he sums this up really well. The perception of venture capitalists is that diagnostic science is risky. There's a very significant risk that things will not go as planned in a startup. And at the same time, these startups have a significant level of capital intensity. So they need tens of millions of dollars to be successful. They're not small investments, they're actually big investments. And maybe one of the biggest problems is that it's difficult to extract value-based pricing for diagnostics. So at the moment, a COVID test is somewhere between $50 and $100. And so the amount of profit that can be realized on that test is very small. And so it's hard to make a lot of money on diagnostic investments. And I've been part of several diagnostic startups that all together, even kind of, swimming against the stream here, have raised about $150 million. And I've been told by venture capitalists over and over again, it's just too hard to make money in diagnostics. And I've accepted that, but I think now we realize that if we don't have better diagnostics, there's an awful lot of money to be lost. And so while the situation that we're in is unfortunate right now, I think it also highlights that we need investment in this area. We need investment in diagnostics. Having good diagnostics, kind of like having a good army to protect a country from attack. And you don't want to put the army together while you're being attacked. You want it there and ready to be deployed when you need it. So my colleague at the Faculty of Pharmacy, Keith Pardee, and I wrote an op-ed that was published in the Hill Times about this idea that we really need a moonshot for new diagnostics if we want to be able to effectively control pandemics. And I think the idea of a moonshot, it makes a lot of sense. We decided as a society that we were going to put a man on the moon, and we did. And the amount of technological innovation and disruption that was required to do that was immense. I think as a society, we could decide that once and for all, we need better diagnostics, and we need to be able to protect ourselves in the event of a viral pandemic. And then let's find the resources and let's not stop until that's done. So in terms of the unmet needs or the way that we could change our current situation, I think there are a few key things or a few types of breakthroughs that would really make a huge difference. The first one is enabling at-home testing. So giving anybody who wants one a device so that they can test themselves for coronavirus without leaving their house. I think that would be absolutely transformative. And then it takes away the volume problem, right? If we can all test ourselves, we're all doing that individually. We're not asking labs to process hundreds of thousands of samples. For the clinical setting, we need rapid bedside point of care detection using, let's say, PCR or other types of technologies, but it needs to be a lot faster, and we need high throughput so that people can be tested serially and often. We also need to be able to watch what's going on in our environment. Are we seeing a resurgence of the virus, maybe on public transit or in some other place? And can we be alerted to that before a pandemic resurges or before there's a second wave or a third wave so that we can do something about it? And so if we had the ability to do this type of testing in these different environments, I think things could be very different. It would be very straightforward to test healthcare workers on a daily basis. It would be possible to test patients in any clinical setting, and maybe especially in long-term care, as frequently as needed. It might even also allow us to let people in long-term care have visitors, because we can test visitors on demand, on the spot, within 5 minutes, know whether they're carrying anything that's problematic. And so we're not asking a vulnerable population to also be isolated, which has a variety of detrimental effects. We would also be better equipped to test in remote areas, test in places like Northern Canada if the outbreak becomes problematic there. As well, we are going to need to really step up our surveillance when people are coming into the country, let's say at airports, border crossings, just so we know whether someone is carrying coronavirus with them, instead of asking people to quarantine, let's say with certainty whether or not there's a problem. So I think these new technological innovations, disruptions, they're needed in order to take us into a new era where diagnostic testing looks completely different. It's not swab, send the swab to a lab, wait for the swabs to be processed, return the test result a few days later, problems with volumes, all the things that I've described to you, but instead just very routine measurements that we can do every day, multiple times a day if we need to. So then with that food for thought, I'm going to shift gears for a few minutes, but I'm going to come back to all of this because given what I've told you about the startup scene in diagnostics, which has been difficult, I think turning to academic researchers and academic institutions to produce the next generation of diagnostic testing technologies makes a lot of sense. And so, not necessarily in preparation for this, but as a foundational move on our part, we formed a group called Prime about a year ago. And this brought together many of our best and brightest at the University of Toronto that are working in the life sciences, people that are working on diagnostic development, but also drug discovery, disease biology. We brought people together from 12 different U of T departments, so really kind of brought people out of their silos so that they were better networked, so that they were collaborating more. And this is a group of people. I'm very proud to be the director of Prime, and we have a group of faculty members and trainees. We have about 100 trainees that are involved in Prime, and this is a group of people with a really exceptional track record. So when the pandemic really took off, what we were able to do at Prime was to convene some of our faculty members that had very relevant expertise for the development of new diagnostics, also new therapeutics for coronavirus. And so the roster of our task force is shown here. And we got together in March. We started brainstorming about new types of technologies that maybe we could fast track and get up and running. We also identified some really critical central resources that people developing new diagnostic tests could benefit from. And so I've listed those on the right-hand side, things like pseudotype viral particles. So these are models for SARS-CoV-2, but are a lot safer to work with. So they're exactly the type of material that you would want to work with to develop a new diagnostic test. And luckily, Ellen Cochran, one of our members, had those ready to go. And so we were able to take advantage of that. Jim Rini, who's another member of our community, has been working on coronaviruses for some time and was quickly able to generate antibodies and also some of the proteins from SARS-CoV-2, another set of essential reagents for diagnostic test development. Leah Cowan also spearheaded the development of a biobank where clinical specimens could be deposited for general use, also specimens of the virus. And then we're very fortunate at the University of Toronto to have a biosafety level 3 lab that was quickly repurposed in order to culture the SARS-CoV-2 virus and also to have animal models of viral infection up and running so that new technologies could be tested using animal models of infection. And so within a very short amount of time, we were able to identify these central resources and also identify people that had really exciting technologies to bring forward to make an impact on the pandemic. I'll describe a few of these to you. I'll start with a couple technologies that are focused on antibody testing. So again, this is testing to determine whether someone has been exposed to the SARS-CoV-2 virus. And again, this is going to help us get a handle on the prevalence of infection in the general community. And this type of testing, it's very well accepted that we need it, but it turns out that many of the tests that are available for this type of analysis are quite unreliable. And so this was a story that came out from the CDC where they analyzed commercially available tests and really found them quite unsatisfactory. So there's certainly a lot of room to improve in this area. And so two of our members, Igor Stagliar and Xinyu Liu, went to work on developing new technologies for this application. Igor has a test that has a few advantages, including that it's quantitative. It's also very, very sensitive, and so allows very low levels of COVID antibodies to be detected. And Xinyu's group is focused on developing point of care tests for antibodies using paper microfluidics. And I also want to mention that these efforts, and the ones that I'll tell you about in a few minutes, were funded by the Toronto COVID-19 Action Council. And this is a fund that was put together by the University of Toronto, also UHN, to quickly find resources for this type of work. And I think this was really an extraordinary effort. It was an incredibly strategic thing to do because many of our federal funding agencies and provincial funding agencies have been relatively slow to respond, whereas this Toronto-based fund was available very quickly, and these projects got up and running in April and May. On the direct viral testing side of things, we have Keith Pardee, my colleague. You may have heard Keith talk about his technology a few months ago on a webinar just like this one. Keith uses synthetic biology to detect viral nucleic acids and uses these really interesting gene circuits as detectors, and has made a tremendous amount of progress over the last few months, and is in the middle of a patient sample trial that will give him enough data, hopefully to get Health Canada approval for his test. We have Leo Chow, who's part of our biomedical engineering group, who's developing a one-step test that's really simple for coronavirus. And then Warren Chan, who's actually the head of our biomedical engineering program at the University of Toronto, has a handheld prototype in development that is based on an interesting type of nanomaterials that emit light as a potential diagnostic for SARS-CoV-2. So really a wide variety of very exciting next generation technologies that we've been able to jumpstart research on by having access to the resources provided by the Toronto COVID-19 Action Council, and also through having an organization like Prime, having the task force, and having people ready to be activated to start working on these very important problems. And so for the last little part of the talk, I'm going to tell you about an effort that is going on in my own research group here in the Faculty of Pharmacy. And this is a different approach from any of the ones that I've told you about so far. And it's an approach that directly targets viral particles. So can we detect viral particles directly? The other thing that's different about it is that we are looking to detect the virus in saliva. So there's been quite a bit of data that has come out over the past few months showing that if you test the saliva of patients infected with SARS-CoV-2, the levels of virus in the saliva are very similar to what you see on a nasopharyngeal swab. So what this presents then is the opportunity to potentially detect the virus directly in the mouth. No samples being collected, but just a quick measurement in the mouth to know whether someone is infected. And so what we have in mind is to develop a type of testing instrument, very small, handheld, really inexpensive, $25, $50, that would allow people to test themselves, would allow testing to be done, let's say, at an airport really quickly, through this type of measurement that is as simple to carry out as, let's say, just taking a temperature using kind of an old school thermometer that goes in the mouth rather than being, shooting some light at the forehead. But any event, really straightforward. And so the tricky thing here is then, how do we develop the type of sensor that would be required to do this type of testing? Because if you think about it, it needs to be totally self-contained. It needs to sit at the end of a disposable sensing probe without any reagents being introduced, because we're doing this in the mouth. And then we need this sensor probe to kind of autonomously report on the absence or presence of the virus in the mouth. And so we've worked out a way to do this over the last few months that I'm very excited about. I think it has quite a bit of promise, but it did start out as really definitely a pie in the sky kind of crazy idea. And the idea was this, that we would build a construct on the surface of an electrode that would report on the presence of viral particles. And the construct looks like this. So here's our sensing surface. So this is basically just a little bit of gold wire. We have a DNA linker that's just a nice rigid tube that's going to help the sensor function. And then we attach an antibody to that linker that specifically binds the SARS-CoV-2 virus. It won't bind anything else. It only binds that type of viral particle. And then we have a redox active molecule that's attached to the DNA linker. And this is what is going to give us some signal to monitor. So this type of approach, we've designed it to be electrochemical because we want the cost of these handheld devices to be very low, and electrochemical sensing it's easy to use, cost-effective instrumentation. And so then the trick here is that in order to detect the presence of the virus, we're going to watch the motion of this construct on a sensor surface. And the way that we're going to trigger this motion happening is we're going to put a potential on the sensor surface that's positive. That's going to attract this DNA linker, which is negative. And so as we put that applied potential on the surface, that construct then is going to travel down to the surface of the electrode, and then this redox label is going to react with the sensor surface. And so how do we use this to detect the presence or absence of the virus? Well, our thought was that if a viral particle is bound to this antibody, that rate of transport will really be attenuated. And that's what we would use as a signal for viral detection. And the reason we thought this would work is if you think about the size of a viral particle, it's enormous compared to this construct that we're using for sensing. And so if we think about an unbound sensor construct that just has our antibody and DNA, that's going to have a pretty fast rate of transport as we're applying a potential. But now when we park a large viral particle onto that construct, the motion is going to be very, very slow as it travels, and it's dragging the viral particle along with it. And sure enough, when we make electrochemical measurements, this is exactly what we see. And so what we're doing here is we're pulsing our potential, we're measuring that electrochemical reaction. If the virus is not present, we see a fast decay of the electrochemical transient. That's what's shown here in this gray dotted line. If we happen to have just a viral protein and not a viral particle, it slows down a little bit. But if we do have the virus present in a sample, that transport slows way down. And we can see that at very, very low levels of virus. So we think this is a solution to enable this kind of probe-based, directly in the mouth type of measurement that could be done very rapidly on the time course of minutes. And so where we're going from here is developing this handheld prototype. We have the sensing mechanism worked out, and so now we're turning to developing the instrumentation. We're working with clinical partners to look at clinical specimens to make sure that we have the needed level of sensitivity and specificity for this to be an accurate type of diagnostic testing. And then again, by accessing that really unique biosafety level 3 facility that we have on campus and have access to, we can actually make these measurements in the oral cavity of rodents to be sure that we're ready to start doing this with humans. So this is all ongoing. And I want to point out the 3 people that are on the bottom of the slide here, Hanya Yosefi, Dinkran Cheng, and Alam Mahmood, who even while all the labs were shut down, were coming in and really fast tracking the development of this and getting this up and running in a matter of weeks. And I really commend them for their efforts. So with that, I want to sum up and hopefully also hear some questions from you all. And so I'll just close with a few points, and hopefully I've convinced you that we really do need rapid on-demand diagnostic tools to combat COVID-19 and to keep dealing with the local outbreaks that we'll see of the virus as shutdowns are alleviated. And certainly, we hope that a vaccine is coming. We don't know when it will come. We may be living with this for quite a while, and we're going to need the ability to very rapidly respond to this pandemic. And then I think it's clear that there's a danger that this may happen over and over again. But the types of tools that I told you about today, we have to find some way to get them out of academic research laboratories and into the clinic. And that's going to require sustained investment in diagnostic technology development. Again, this is an army that we want standing guard so that it's there when we need it. The new technologies that we develop, we really need to make sure that they're quite adaptable. Right now we're dealing with SARS-CoV-2, but it could be something different in a month or in a year or in 10 years. And so we need things to be quite adaptable and to be tools that we can quickly get up and running. And we really have to keep an eye on this point about vulnerable populations. Anything that we develop, it shouldn't necessarily be a replacement for lab-based testing. It should be something that really helps us to better protect the people that are most vulnerable. We need widespread, rapid, and cost-effective testing. But most of all, we need to protect people that are most at risk when this type of pandemic emerges. So with that, I want to thank you all for your time. I really appreciate you joining this webinar. And I'm very happy to answer your questions. So there's an interface for you to type in questions, and then they'll come through to me. I'll read the question and then do my best to answer them. And so please, any questions, comments, feedback, and don't hesitate. The questions can be big, small, related to what I talked about, maybe not so related. Please just send over anything that you'd like to use the remaining time to address. And so I'll start with the first question that's come across here to me. So this question says, from the media reports, it sounds like the virus keeps changing. So how are we going to deal with this? How can we develop diagnostics to address mutated forms of the virus? How is this going to work? So this is a great question and something that absolutely has to be kept front and center as we develop new diagnostics. What we typically do is we try to target parts of the virus that are not known to mutate. And it's a little hard to know this in advance, whether or not a part of the virus is going to mutate on you. But just through bioinformatics and analysis of the genome of the virus, you can usually hone in on something that won't change. And so most of the diagnostics tend to latch on to RNA sequences or protein sequences where there shouldn't be mutational hotspots. In the case of the system that we're developing, we're actually targeting the virus through viral proteins that are essential. If the virus doesn't have these proteins, then they're not going to be able to infect human cells. And so in that case, we have a pretty good shot in terms of being able to deal with mutated versions of proteins. Okay, so here's another question. Novel diagnostic tools, therapeutics, and emergency preparedness are great. However, should we also expect more investment for policy changes? Since many countries proximate to the origin of COVID, such as Taiwan, South Korea, Hong Kong, Japan, have mature public health infrastructure and trained care providers, and they've yielded better results than North America and the EU, although the latter regions traditionally have more robust biotech landscapes, academic organizations, and federal funding. So this is a great question. You know, what is it really just a matter of putting more money into diagnostics? Well, no. I mean, it's a matter of having a robust biotech ecosystem, whether it's diagnostic development, whether it's therapeutic development, whether it's vaccine development, so that we have all of these tools nationally. And I think this is really critical. Some of the countries that did very well, and South Korea is a great example, they gathered up a bunch of local companies in South Korea and said, show us what you have. And the platforms that those companies had, then they scaled them up and used them to really suppress the outbreak. In Canada, we don't have that ecosystem of small companies. It's been very difficult to get companies up and running and to sustain them. And so I think it's about really having a robust ecosystem, which would be great for us economically, but it also is absolutely critical when we look at these types of pandemic outbreaks and all of the implications. We need this. And then, you know, in terms of investment and policy changes, all of these things, maybe that's a little bit outside of my area of expertise, but I think, you know, all of us have changed our perspective over the last few months in terms of the role of health and health technologies and how important these are just for us to be able to live our lives, not necessarily for the extension of life or dealing with chronic diseases, but just to be able to live in the face of a viral pandemic that honestly could have been a lot worse, right? In terms of the lethality of the virus, we could have been dealing with something much more deadly, which would have been, what we've dealt with has been incredibly problematic, and there's been way too many lives lost, but it could have been quite a bit worse. Okay, here's a question. How soon do you think antibody testing will be widely available, and what type of distribution do you see? So this is a great question. I would love to take an antibody test myself, and they're not really around yet in a form, I think, that the health agencies trust. There has been some incredible work that has gone on at the Lunenfeld and Mount Sinai down the street from here, where they are developing a test that can be run in their laboratories that is giving really good results, very accurate. So I think this may be our shortest term hope in terms of getting those antibody testing results back to people. The time sensitivity is a little bit less acute there, right? If you needed a week of turnaround time, that's okay. You're trying to give somebody information about their status. And then we also need to decide as a society how we're going to treat that information once we do tell people whether they have COVID antibodies or not. Okay, here's another question about the antibody test. The World Health Organization does not recommend the antibody test for TB. Can we use it for COVID? And I think this is referring to antibody-based tests if you're using an antibody to actually catch, let's say, a tuberculosis cell or a viral pathogen. And I think the important thing here is that you can't say that one type of test doesn't work for all indications or it does work for all indications. It's very specific to the specific antibody, to the levels of the pathogen in a clinical sample. TB is a very tough type of organism to work with. It's typically in the sputum, which is difficult to work with. COVID is relatively easier to deal with. it's in the saliva, it's in the nasal cavity. There's a lot of places to access it, and the loads are pretty high. In terms of the antibody-based tests, in case that was the question, it really depends what information you're trying to get at. If you're looking for active infection, you probably don't want to use an antibody-based test where you're actually looking at antibodies as a biomarker. It's much better to look at the pathogen directly. Okay, and then there's another question about the limit of detection for our point-of-care electrochemical system that we are developing. So what we are aiming to do, and we have some very good results so far, we really want sensitivity that is the same as the PCR-based test. I know from past experience that if your sensitivity isn't very good, clinicians are not going to want to use the tool you develop because you're going to have a lot of false negatives. And so we are aiming to be around the 10 to the 3, 10 to the 4 viral particles per mil level, because that's where the molecular tests are. And so we have a few strategies for that, because I think maybe what you're alluding to, just based on the first part of the question, is that in the past, antibody-based methods where you're using an antibody to catch a pathogen have not been that sensitive. But here we have a surface loaded with antibodies. We can use multiple antibodies if we need multivalent interactions to give us a higher binding affinity. And so we have a lot of degrees of freedom that may not have been present in other systems to give us the needed levels of sensitivity. Okay, here's another question. If we are only testing for virus in saliva and nasal swabs, we are missing out if other organs are infected by COVID. Any thoughts on how we can work on that? Yeah, that's a tough problem. The specimens that we're most readily going to test are the ones that we can access, and organ infection is a tough one just because of access to the organs. And so some of that work may need to be done, let's say, on a post-mortem basis. This is also where the animal studies may be incredibly valuable, that we can get at that type of information in an animal model, form a hypothesis, and then look in humans. But it's a very important question. One of the reasons that the COVID pandemic has been as unwieldy as we've experienced it is because of all of these different presentations, right? A viral infection. It's not always just a cough and respiratory infections. There are a variety of other presentations. And so getting at the physiology behind that is absolutely critical. So I don't have a great answer in terms of something that we're working on, but I think there are quite a few people that are diving into that. Okay, so I think those are the burning questions that people had. Again, thank you so much for joining us. We really appreciate your time and attention. And I hope everybody is doing well, staying safe, and we look forward to having you attend